loud, and hopefully this will come up. Oh, hey, that's perfect. Awesome. Yeah. Yes, it's all good. All right.
Great. Yes, my name is Janelle Hare. I'm at Morehead State University. I'm a biologist. And you can see the very long title of my grant that I'm kind of stuck with. What I like to think of it in short is how Acinetobacter controls its responses to DNA damage. I've been fortunate. At the bottom, you see my acknowledgements to be supported by a variety of sources, especially lots of support from INBRI for postdoc awards, bridge awards, some next gen sequencing awards, all sorts of great stuff. So I really appreciate that. In my lab, we study a soil-loving, a hospital-loving, gram-negative bacterium called Acinetobacter baumannii. You can see it in the background. And you can see the hospital view in the upper right, using UV light to disinfect the hospital setting. Well, Acinetobacter baumannii is an opportunistic pathogen. So it's present in the hospital. It's very hardy as an organism to dryness, and it is antibiotic resistant very strongly. And so that makes it a big problem for treatment. What we study are the responses of these bacteria when their DNA gets damaged, such as you might think with exposure to radiation like UV light, which is actually one of the things we use in our lab to cause DNA damage, is we expose the cells to UV light. So mostly I'm going to talk about the actual genes that cause regulation of other genes. So very early in the process, not as much what happens after they instigate the program, but how do they know that DNA damage is around, and how do they start recruiting the actions of other gene products to cope with this problem? So our goal is then to learn how this pathogen works. We see in green some of the triggers of this so-called SOS, cry for help response after DNA damage. Antimicrobials can cause this, UV light can cause this, many other things. And then the problem is this can lead to the buildup of antibiotic resistance in these bacteria, can lead to virulence factors that can help them cause disease, and even the transfer of these activities to other bacteria. So it's a big, wide problem. I'm going to show you in the next few slides kind of the canonical model that people have developed in bacteria like E. coli, Staph aureus, and here we're seeing the default position in the absence of DNA damage. So there's a protein, let's say, that forms a dimer with itself, and it binds to these target sequences in the promoters of genes called SOS boxes, and this blocks transcription of these genes, all sorts of genes, many, many tens of genes. But after DNA damage, there's usually some single-stranded DNA in the cell, and that can bind to a protein called RecA, which then changes its conformation. It binds to LexA, and it helps LexA carry out a cleavage event on itself. And when LexA has been cleaved, it dissociates from the promoters, and we're able to get transcription of mRNA, and then we can get gene products translated from that mRNA. So these gene products down at the bottom are some examples of typical ones, again, from some of the highly studied models. I'm not going to go into detail what these kinds of proteins and genes would be rather for our organism, but INBRI did fund a next generation sequencing project to look at the whole transcriptome of all the genes that were responsive, either up or down after DNA damage. And so that was quite a while for us, but of course there was a lot to go through and work through with the students that could keep us busy for probably the rest of my career. What we study, not necessarily the actions of these genes, because these genes are the ones that are trying to fix the problem without any errors. They're cutting out bases, they're cutting out pieces of DNA, they're recombining within the cell to try to get things back to the way they were before DNA damage. But sometimes that doesn't work. And when there's too much DNA damage going on for too long, we see some other genes that are being expressed, that are able to take a different approach to the problem, but in doing so, they cause mutations. So these sorts of proteins are called UMUD and UMUC. They're also induced after DNA damage, and like LEX-A, UMUD cleaves itself and interacts with UMUC and forms a polymerase that can replicate damaged DNA. But it's got an extra large active site as an enzyme, and so it will misincorporate some bases and cause some mutations. And we call that process SOS mutagenesis. So mutations arise. The part of this process that we're getting closer to what we're interested in my lab is the UMUD protein. So that's kind of where we're going to start diving in soon. So this is what we know happens in a lot of bacteria, but not in Acinetobacter in quite the same way. You know, Acinetobacter don't even live in our body like E. coli and some Staphylococci do. They are experiencing different evolutionary pressures, and over time, they have different processes that they're undergoing. For example, we first noticed that not the pathogen, but a different species in this genus has a different form of this UMUD gene. It's extra large, it has an extra N-terminal region at the beginning, and it's next to a screwed up UMUC gene. It's truncated, it's missing most of itself. So this ADP species cannot do SOS mutagenesis. But the pathogen that we've started studying more and more, Acinetobacter baumannii, shown on the right, not only can do SOS mutagenesis because it has UMUD and UMUC genes, but it has many, many copies of UMUD and C genes, like 4 copies of UMUC, 2 copies of UMUD, and in addition, this weirdo UMUD AB gene. So, you know, what the Acinetobacter share, both of these species that I've told you about, is that they don't even have a LEX-A gene in this organism. Now, we do know that they do have some sort of response because some of these species can do this SOS mutagenesis response process, but there's no LEX-A there. So leading to our question, how do the bacteria in this genus respond to DNA damage without having a LEX-A protein? What do they use in place of it? Well, what we know is that there are more than just UMUD and UMUC and UMUD-AB, all these genes. It's named for UV mutagenesis as a gene name. We know that there are many genes that are induced after DNA damage, meaning that increase their expression, their transcription after DNA damage. Right next door to UMUD-AB that we're starting to study is this gene I've shown here in purple, DDRR. It is, we look down at the bottom left, highly expressed after DNA damage. So for some reason, the cells want to make more of it after DNA damage, probably has some role. And actually, all the genes on this slide are induced after DNA damage. All of these polymerase genes and UMUD-AB and DDRR. So we're trying to figure out how these genes are working together. If they're working together, what are they doing to provide an adequate response so these cells can stay alive after DNA damage? The end game is that now we know that both this DDRR protein and UMUD protein produced from this upregulation of transcription are regulatory proteins that are used to control some, but not all of the genes that are responsive to DNA damage. So it's a specialized sort of response that's going on in the cells. So when I came to MSU about 19 years ago, on the right, it's kind of the knowledge base of what we knew about UMUD-AB. We knew some things in the blue column about what LEX-A proteins in other species do. We knew the same sets of information for what UMUD proteins in other species do, but neither one of those were present in our Acinetobacter bailey that I was starting to study. And so it's like, well, what is UMUD-AB? Is it like a LEX-A? Is it a UMUD? Is there something that it's doing that is providing a response? Is it responsive to DNA damage? So we looked at some of the phenotypes kind of shown in the middle of this table. I'm not going to go through all of them, just kind of looking at the color coding. We went through some of the biochemical properties that UMUD-AB might display before and after DNA damage that might help us understand a function for it in the cell. If it was like LEX-A in regulating, or if it was like UMUD in working with a polymerase, what was it doing? So what we found out was that in many cases, most of its behavior was like that of a LEX-A regulatory protein. The way it cleaved itself and the needs that it had, the part of the protein that were important, all of these seem to, well, most of them seem to be like a LEX-A regulator. So we thought, well, let's check and see if UMUD-AB is actually regulating DNA damage-induced genes. And we found that it was, that only some of the induced genes after DNA damage were regulated and repressed specifically by UMUD-AB. So the ones shown on the left-hand side of this graph. This graph is looking at genes being expressed in the absence and then the presence of DNA damage. So within each color pair, the first lighter color is without DNA damage. The darker color is with DNA damage. And all the orange-red are wild type, showing greater production of these genes after DNA damage. Well, we see that there's about 8 genes shown here. And for the genes on the left-hand side, all of our genes were dysregulated in the UMUD-AB mutant. So the blue lines represent what we saw for these genes being expressed in a UMUD-AB mutant. So that was a surprise that it was actually repressing the activity of these genes. On the right-hand side of the graph, though, we realized that UMUD-AB wasn't in control of all the genes that were responsive to DNA damage. Only this small group of UMUD and C error-prone polymerase genes and itself, UMUD-AB, and DDRR. So we wanted to understand how it's working as a repressor. So we dug into that a little bit more. And I had a student come up to me, Travis, as you see there. He said, oh, hey, Dr. Hare, I looked online and I found a computer model program that has produced a potential model for the structure of our UMUD-AB protein. And it looks a lot like LEX-A. I'm like, oh, that's so cool. Let's take a look. And so if you look at the kind of bluish cyan portions on the left side of each of these proteins, you'll see that they have a similar kind of triangular predicted configuration. And so based on that, we thought, well, if our protein adopts the same shape, maybe it's doing the same things with that part of the protein. And so he and other students in the lab created some mutants by substituting proline residues in for those important regions in the helix, just breaking and disrupting the helix. And we saw that in those mutants, the cells could not repress gene activity anymore or gene transcription. So those helices seem to be important for maybe interacting with DNA. Now, there are some other groups that have actually shown that UMUD-AB does bind to DNA. We haven't done those studies, but we do know as a scientific community that UMUD-AB does bind DNA. We also, on the right-hand side, looked to see what was required for UMUD-AB to cleave and then allow genes to be induced, because there are 2 parts to its action as a regulator: repression of gene transcription in the absence of DNA damage, and then relief of repression when the protein cleaves itself, producing, the ability to get transcribed. So then we started to fill in more of our table and realizing that UMUD-AB seems to be a regulatory protein. It uses this helix region in the N-terminus of the protein to do the regulation. And so we could move on from there. And there it got really even harder. The mystery being, what does DDRR do in the SOS response? It's the only gene besides the regulator UMUD-AB, besides all the UMUD-C proteins. So seems like it's involved somehow. What is it doing? Ran into a little bit of a black box though, of course, because there are, unlike the previous work I've told you about, there are really no homologs in other species. It is unique, as we see down here, to the genus Acinetobacter. It's a small protein. It's always next to Umud-Ab. So they're always conserved and present in the chromosome of these bacteria at the same place. So for some reason, evolutionarily, seems like they're possibly working together or conserved to be in this unit together. So there were some obvious possibilities. One is on the left-hand side that maybe DDRR could help UMUD cleave itself, because we know UMUD-Ab has to cleave itself properly to get the proper control. On the flip side, maybe we thought DDRR could help UMUD-Ab bind to DNA and do its repression action. So which of the two major actions of UMUD-Ab in being a regulator was there room for DDRR to participate in? That was our main initial approach. What we found is that DDRR represses the same set of genes as UMUD-Ab. If UMUD represses it, DDRR represses it. If UMUD-Ab does not repress a gene, DDRR does not repress that gene either. So they really seem to be working together. Here's the same sort of scenario where the orange and red are all of the wild type responses showing increased expression after DNA damage. And here in blue are the responses of the DNA of the DDRR mutants. So we see just completely off the charts high expression of all these genes if you mutate DDRR. So it too is required for repression. Drilling down a little bit further into that, what we've been doing most recently with a student, you can see down at the bottom right, Belinda last spring made a mutant in which UMUD-Ab could not cleave itself. And you may say, well, didn't you already show us these data? You've already known this. Well, we knew it in a different species. And in order to proceed, we had to first establish that in the pathogen Baumannii, these same findings held true. So she made the mutant, and indeed, we saw that if you can't cleave UMUD-Ab, you cannot induce genes. So just 2 of the about 8 shown here, they all work the same way. So I'm just showing you a few of them. So whereas we would expect here in the wild type an increase in production of gene transcription after DNA damage, you don't see any increased gene expression if you mutate this one amino acid that's required for the protein to cleave itself. When we added, though, a DDRR mutation into that background, we saw again that DDRR mutant cells were unable to repress DNA gene transcription in the absence of DNA damage. So this again suggested that DDRR helped something about the system repress gene transcription. And most recently, my postdoc, Deb Cook, has been working on many things. One of them is this project, doing some Western blotting, looking at the actual activities of proteins in the living cells. So on the right, we see some immunoblotting where we're applying antibodies to UMUD-Ab, and we're seeing if it's intact large size or if it's been cleaved to small pieces. And on the left and the right, in those very colorful blots, we see that both wild type and DDR mutants are able to have UMUD-Ab cleave itself. So we see, first of all, a green band at a high molecular weight, and then over time, after we've applied DNA damage, we see smaller fragments appearing that represent the cleavage products. And when we kind of look to see how long it took for these 2 strains to cleave UMUD-Ab, it really looks like they are progressing along the same sort of time scale, under about 20 minutes. We've done this several times. We get slightly varying time periods, but we don't really ever see any difference if we mutate DDRR. So DDRR is not really seem to be involved in cleaving UMUD. That kind of leaves open the idea that it's helping with this N-terminal region, maybe that is causing the protein to bind and repress. What we found when a different student, Molly, made some mutants also last spring, is that these two helices are both required and important to repress transcription, but helix 2 is kind of more important. So when we mutate helix 2, we see even greater derepression of gene transcription. So we're trying to find a little bit more out about the different roles of the different parts within this protein. And yes, sure enough, we then applied the DDRR mutation into that strain background, and we see, again, further increase in the transcription of these all the same genes. So what we're thinking this probably means is that DDRR doesn't seem to be doing exactly the same thing as these two helices, or else when we mutated it, we wouldn't see any increase in expression because the mutations to the helices would have already ruined whatever the helices were doing. So we're seeing an additive effect, meaning there's perhaps something different that DDRR is doing with UMUD to repress these genes. So that's kind of what we're trying to dig into these days. And here's my last slide. We are focusing now, moving into working on another R15 grant, where we have kind of two main parts to it. One of it is trying to see if the way that these two proteins repress together involve them binding together, co-binding, not really a term, but do they physically touch each other? Because our most recent paper last year wasn't able to see them actually binding to each other in two hybrid experiments, but in those sorts of experiments, you're taking your protein and you're hooking it to a bigger protein and you're making a fusion protein. And it's possible that that could have interfered with their ability to interact with each other. So we're taking some different approaches in the new R15. And the thing that's I'm pretty excited about is that we've noticed some other phenotypes of cell behavior when we mutate DDRR. And they seem to be related to inhibiting cells dividing after DNA damage. If you look down at the bottom at the pretty microscopy pictures, you can see that here on the left, that cells become long after they've been damaged by DNA because they don't want to divide. They want to wait until they fix their problems and then divide. So wild type cells will become very long after you treat them with DNA damaging agents. But that doesn't happen in the DDRR mutants. Somehow they're not catching on to the fact that they're being DNA damaged and that they should wait and decide to divide later. So we're trying to understand if that's a direct role of the DDRR protein or maybe an indirect role where it's possibly working with another regulator to control additional sets of genes besides the ones we know it works with UMUDAB on. So we just are pretty excited about this new phenotype opening up. I've recruited some more students to the lab. I've got about 5 students right now working with me. Molly and Belinda are as well. And some craft undergrad, they're not even undergraduates, craft students that are in high school are starting to work on the project too. So it's a pretty exciting time. I thank you for your attention and I'd be happy to take any questions.
Great, thanks, Janelle, and congratulations on the R15. We're
much.
a little behind schedule, but maybe have time for one question, if anybody. I was just curious about, you had talked about at the beginning that the bacteria can transfer some of this resistance or whatever to other bacteria. How does that come about?
Well, some of the genes that are induced are located in a bacteriophage region of the chromosome. And we showed in some of our early work that there are actually bacteriophage virus particles produced after DNA damage, which is pretty common in the field. So we think that probably when they're stressed by DNA damage, you get packaging of the virus DNA into the virus particle and that it can go and donate that DNA to new bacterial hosts. The problem is that some of these error-prone polymerases are encoded in the bacteriophage. So what they're carrying with them is the ability to make a mutagenic polymerase. That's the hypothesis. Yeah,
that's cool. Okay, well, we better move on unless anybody else has a burning question.
Nope, we got to move on. Thanks.
Simran, can you
Yes,
share your
I'm
screen?
here. Yeah, I'm here. So I'm having an issue.
Yeah, it should, if you hover your mouse,
Yeah.
you see the,
Yes, I know. And I'm trying to say share and it doesn't allow me. And I've done this all the time and I don't know what's happening today. My computer has been giving me little issues and I actually had to shut it down completely. But I don't know what is going on. I'm so sorry. We already behind and I'm like,
well, yeah. Well, if you want, we could move on to Melissa and then come back. If I don't.
that would be okay. I'm trying to figure out what's going on here.
Okay, is that okay with you, Melissa?
Yeah, sure.
All right. So you.
Okay, are you seeing my slides?
Yeah, yeah.
Okay.
Looks
Got
good,
it.
thanks.
I had some issues earlier too. So I'm Melissa Mefford. I'm in my 4th year as an assistant professor in the Department of Biology and Chemistry at Morehead State University. And in the Mefford lab, we're interested generally in chromosome structure, but more specifically in the ends of linear chromosomes, which are specialized regions called telomeres. So in the lab, we have 2 projects going on looking at the evolution of telomeres and also the enzyme telomerase that helps to regulate them. So a bit of brief background on telomeres. They're repetitive DNA sequences located at the ends of linear eukaryotic chromosomes. So you can see them highlighted in blue on these chromosome stains below. This repetitive DNA sequence is bound by proteins, and together these provide protective caps to the ends of chromosomes. We often use the analogy that they act like aglets, or those plastic tips at the ends of shoelaces, to protect the ends of chromosomes. So I'm sure we've all had a favorite pair of tennis shoes where we've lost that plastic tip and the ends begin to fray. So telomeres help protect chromosomes from getting degraded from the ends. So while they do provide positive aspects to the cells that contain them, telomeres also present the cell with a challenge, which is that when the DNA is copied during DNA replication, the ends can't be copied by the normal DNA replication machinery. And we refer to this copying issue as the end replication problem. So I'm showing the end of a telomere here that needs to be copied or replicated. So the original DNA strand is going to separate and the new strand is going to be copied from that template in the 5' to 3' direction. So on one of the strands, this happens in a continuous fashion. That's the leading strand. However, on the bottom strand, the lagging strand, the DNA has to be synthesized in small fragments, and it requires an RNA primer shown in magenta, which are ultimately degraded. So once that terminal RNA primer is degraded, it leaves a portion of that new strand that wasn't copied. So over time, chromosomes would continually get a little bit shorter on the new lagging strand pieces. So to overcome this end replication problem, most eukaryotic organisms express an enzyme complex called telomerase. Telomerase minimally requires 2 components, a telomerase RNA component, which provides a template that specifies the particular nucleotide repeat sequence to be added, and then a TERT reverse transcriptase protein, which catalyzes the addition of nucleotides. So telomerase is going to add nucleotides to the original 3' strand, making it longer and allowing space for one of those new Okazaki fragments to be laid down. So we know that loss of telomerase activity is going to lead to telomere shortening. And when telomeres reach a critically short length, it causes a cell cycle arrest known as senescence. We also know that telomeres and telomerase are implicated in 2 very important human health issues, which are aging and cancer. So in humans, for instance, most cells actually don't express telomerase, and so their telomeres do shorten with aging. And we also know that in cancers, the cells need to be able to replicate and divide uncontrollably. And so over 85% of cancers upregulate expression of the telomerase enzyme. So in my lab, we're interested in some very basic science questions of if linear chromosomes and telomeres create this end replication problem, then why did linear chromosomes, telomeres, and telomerase evolve? So we know that in bacteria or prokaryotes, they contain a single circular chromosome. So we have two projects going on in the lab. Our first project or specific aim is to genetically engineer circularized versions of linear chromosomes in a eukaryote. And I'm going to focus on that project today. I probably won't have time to tell you about our second project, which focuses more on the telomerase enzyme and specifically looking for gain-of-function mutations in the telomerase RNA component. So to do these projects, we take advantage of a very powerful model organism called Saccharomyces cerevisiae, or more commonly called budding yeast. So budding yeast is single-celled, but it is eukaryotic. It is cheap, quick, and easy to grow in the lab. It has a sequenced and very well annotated genome, including in the telomeric and subtelomeric regions. It also has an extensive genetic toolbox of tools that we can use to easily manipulate the genome. It also exists in both a diploid and a haploid state. So the diploid contains 2 copies of all chromosomes like humans. Haploids contain 1 copy of all chromosomes, and those are the strains that we generally are working with in the lab. Makes doing genetics easier. But yeast do have a complex life cycle where 2 haploid strains can mate to form a diploid, and the diploid can undergo meiosis to form haploids. So it has both asexual and sexual reproduction. Budding yeast have 16 chromosomes and therefore 32 telomeres. And they are also useful for studying telomerase because these single-celled organisms are constantly dividing, and so they constitutively express the telomerase enzyme. So again, our first project in the lab is aiming to circularize these linear yeast chromosomes, which is going to delete the telomeres and allow us to ask whether the telomeres are doing anything important. And it's again, getting at this big, broad question of why did linear chromosomes evolve if it creates this significant end replication problem. So the approach is to genetically engineer strains of Saccharomyces cerevisiae with circularized versions of their linear chromosomes. And the long-term goals of this project are to create 16 different strains with each of the chromosomes individually circularized. And that's what we're currently working on. If these circularized chromosomes are viable, then our longer term goals are to combine those circularized chromosomes to create a strain with all but 1 circular chromosome, which would allow us to study single telomere dynamics, which has been a challenge for the yeast telomere field. And then also to create a strain with all 16 chromosomes circularized, which would allow us to study some controversial extracurricular roles of telomerase, as well as to test experimentally some theories of linear chromosome evolution. And some folks have perhaps called this project crazy, that you can't make a eukaryote with circular chromosomes, but there is some previous data from yeast that suggests that this would be very possible. So back in the 1980s, during a mutagenic study, a group isolated yeast that contained a single circularized version of their chromosome 3. In the 90s, folks working on a distantly related yeast, Schizosaccharomyces pombe, found that when you deleted some telomere components, that the telomere shortened and that the 3 chromosomes in pombe circularized at a small fraction. So it was an example of a yeast with a fully circularized genome. And then more recently, there was a group that took our yeast, Saccharomyces cerevisiae, and they first fused the 16 linear chromosomes into 1 gigantic long chromosome, and then they followed that up by circularizing that singular chromosome. So there is evidence that Saccharomyces cerevisiae can survive with a solely circular genome. The approach that we are taking is different than all of these studies that I cited, because we are taking an approach that should not include a additional mutations that some of these other studies have had. So I think our approach is different and could add something to this question. So to briefly go over the approach that we're using to circularize our chromosomes, our first step is to build 2 chunks of DNA that we call DNA cassettes. One chunk we're going to insert into the left arm of a chromosome. The other chunk we insert into the right arm of the chromosome. And these cassettes contain several important bits of DNA. So first off, each cassette contains a unique selectable marker gene. So we have a His3 gene for the left arm and a Lu2 gene for the right arm. This allows us to detect whether these cassettes have gotten integrated into the chromosome arms. The other important thing that they contain is half of a third selectable marker gene. This is a gene called URA3. So we put the 5' half of the gene into the right arm cassette and the 3' end of the gene into the left arm cassette. So since this gene is split into 2, it's not functional, but we have shown by the striped area a region of homology that will allow that gene to recombine and form an intact version that is going to be important in our last step for selecting for circularization. And then the last thing that we add to the cassette is shown in green. We have a bit of homology to the particular chromosome arm that we want these cassettes to go into. So once we build these DNA cassettes using PCR, our next step is to put that DNA into our yeast cells and allow the DNA to integrate into the left and right arms of a single chromosome. And we can tell whether that's happened by using selection on growth media lacking either histidine for the left arm or leucine for the right arm. So once we've got a strain that has both of these cassettes integrated into the chromosome arms, then we can allow recombination to occur between that overlapping region of the URA3 gene. When that DNA recombination event happens, it is going to create an intact version of the URA3 gene, and it's also going to cause loss of the telomere terminal regions, including the Lu2 and His3 selectable marker genes. So just last year, we had our first successful circularization of one of the 16 chromosomes. It's chromosome number 4. So I'll show you some of the data that shows our success. And I'll start with some growth data. So I'm going to be showing yeast grown on different types of media along the bottom. And the 3 slices contain 3 different strains. The top slice contains our wild type parental strain. The middle slice contains our double integrant strain, so the one that has both cassettes inserted in the linear form. And then the bottom left slice is going to show the circularized version. So on rich media, which you're looking at now, all of these strains are able to grow. However, on our various selective media, we see that on media lacking histidine or leucine, only the double integrants with the cassettes inserted are able to grow. And on the right, on media lacking uracil, only the circularized chromosome that has recombined the URA3 gene is now able to grow. So to confirm that we have circularization, we have also done PCRs with various sets of primers on genomic DNA isolated from these 3 strains. So on the top gel, we have PCRs from the wild type parent, and the middle gel are PCRs from the double integrant, and on the bottom gel are PCRs from our circularized strain. The cartoons on the left use brackets to indicate the location of the various PCR primers. And for the sake of time, I won't walk you through every single lane and band on this gel, but I'll draw your attention to the band in red on the bottom right. So in our circularized chromosome, we have primers that span the circular junction that should only give us primers if circularization of the chromosome has occurred. And you see that we do see a band of the expected size that's only present in our circular strain. So this suggests that we do in fact have joining of the ends of this chromosome 4. So this was very exciting and was the first evidence that this approach is actually viable and working and can circularize a chromosome. So now that we've got a circularized chromosome strain, what are we going to do with it besides say, look what we did? So we want to characterize the relative fitness of this circularized chromosome strain versus wild type. So we've done that through several different growth assays. So by growing them on media, we can look for differences in colony size or morphology, and we don't see any differences. We can also look more closely at doubling time, which we can quantitatively calculate in liquid culture, and we also do not see any significant differences in doubling time between our circularized strain and wild type. We can also use various approaches to try to challenge the cells and see if the circular chromosome is deficient under more challenging conditions. So thus far, we've tested various temperatures, both colder and hotter than yeast like to grow, and we don't see any temperature sensitivities either. So for all of the assays we've done, our circularized strain seems to be just as fit as our wild type linear strain. We've also begun looking at cell morphology and budding, which is asexual reproduction. And this is still somewhat ongoing, and we're trying to be quantitative with this, but just by eye, we don't see any obvious defects. We're also working on testing sexual reproduction. So we're working on mating our circularized strain with a linear strain, and then seeing if those diploids can then undergo meiosis to form spores, which we call sporulation. This is also ongoing and in process. The cells seem to mate just fine, and we're at the step of sporulation currently. And then another interesting hypothesis I'm excited to test is whether this circularized strain might cause defects in gene expression regulation, as there's emerging evidence that the telomeres help to anchor chromosomes in specific locations within the nucleus, and that's important for regulation of gene expression. So we are in the planning about to get to progress stage of those experiments. Chromosome 4 is not the only chromosome that the lab has been looking at, though it's the one that we've gotten the farthest on. So here I have the 16 chromosomes in yeast lined up with their relative sizes indicated. So we just on Monday this week confirmed that we have successfully circularized a second chromosome, chromosome 8. So these two circularized chromosomes are indicated in dark gray. And somewhat disappointing, though still interesting news, we have 4 chromosomes that we have gotten integration into both arms, and we have tried to circularize them by getting them to grow on -Ura media. And despite a lot of attempts, we cannot get them to grow, so they seem to not circularize. And there's a very interesting question of why some chromosomes will circularize and others won't using this approach. And interestingly, chromosome 3 in cerevisiae has previously been shown to be viable as a circle, is one of the ones that we can't get to circularize. And so we have hypothesized that perhaps where you try to circularize the chromosome is perhaps a very important aspect. And then we have 2 other chromosomes that students are currently working on, where we have one of the integrations confirmed and we're working on the other one. And then we have 8 chromosomes left to start. So very excited about this project. As I mentioned, we also have a second project ongoing in the lab aimed at trying to identify gain of function mutations in the telomerase RNA. But for the sake of time, I'm not going to go into details. I'd be happy to ask any questions. We're making good progress on that project as well. So I'll just quickly acknowledge my funding sources. So I received a new faculty startup award from Cabrin with matching funds from Morehead State. I've also received 2 IDEA awards from Cabrin, and during that time I applied for an NIH R15, which I submitted in February of 2021. This was my very first R15 submission. In July, I received an overall impact score that I was very excited about, and then just found out in January, like 2 days before the funding started, that it was actually funded. So I'm very grateful for Cabrin for their initial support and preparing the IDEA grants. and having the time to collect preliminary data was absolutely key to getting this R15, I think. So thank you for all of the help and feedback. And then finally, thanks to all of the undergrads who have worked in my lab so far. So I'd be happy to answer any questions if anyone has any.
Thanks, great presentation. And congratulations on the R15.
Thanks.
And I also like your little Mefford lab. Now I understand it. The little logo.
Yeah, a telomere and a budding yeast.
And a little budding. So yeah, again, a little behind. Any questions for Melissa? That was really interesting. I think it's particularly interesting that you have these chromosomes that won't. So, I mean, you know.
Yeah, I was about to give up on the project because we actually had the 4 that wouldn't circularize, and then chromosome 4 circularized. And so I was seriously about to scrap this project, and then it became
so I mean,
again all of a sudden.
Probably some cool information in there that why they won't.
Yeah.
All right, we better move on. Simran, do you think you can share your slides now?
Yes, I am going to try again, and hopefully it'll work. No, it doesn't allow me, but what if I send you my slide?
Okay, you can do that, and then maybe we can go on to Lauren. Well, that's
okay. So
because it might take a while. I did finally get Janelle's when. Oh wait,
okay.
no seminar. Okay, no.
I sent you already. If it reached you, I don't know.
Oh, that's the agenda. No, it hasn't come in yet. Sometimes it just takes a while, especially when there's a file attached. Do you mind if we go on to Lauren and then we'll come back?
No, that's okay.
You'll be the final.
I know. That's all right.
Yeah. I was just curious as I was, you think you could see the circularized chromosomes with EM? I'm always pushing EM.
That's a good question. I'd have to look into it and think about it. My initial answer is probably not, but you know, if we could, that'd be great. We've been considering doing fish fluorescence and C2 hybridization.
Oh. Oh, that'd be cool. Okay. So sorry. Yeah. Lauren, you think you have any issues sharing? All right.
All right. We did it. Hi, everyone. I'm Lauren Williamson. I'm also in my 4th year. I'm at Northern Kentucky University. And the mouthful of the title is I've been looking at maternal helminths as a possible anti-inflammatory intervention in a model of neonatal infection. There we go. So the main goal in most psychiatric work and also in my lab is looking at reducing neuroinflammation in prenatal and perinatal periods. So also a total side note, everyone else will be talking about mostly single-celled or very small creatures today, and I'll be talking about rats. But there is bacteria involved with the rats. So the brain does a lot of development very early in life. Rats are a particularly good model because their first week to 10 days of life is a lot like the 3rd trimester in humans. They're born altricial. They do a lot of development. When we are looking at inflammation in babies, we are often looking at making mom sick or giving inflammation at different times along sort of the critical window of brain development that then may cause this is dysbiosis in particular related to all sorts of non-communicable diseases, but also is very correlated with mental health, that depression, anxiety, and other types of psychiatric disorders have inflammation early in life as one of their at least correlated etiologies. So what are some ways we know how to reduce inflammation? So some of the most obvious ones that people have done, and there's tons of literature out there on maternal exercise. So while mom's pregnant, after she's had baby, encouraging exercise in animal models tends to predict and cause better outcomes in babies. Changing maternal diet, so reducing certain types of fatty acids and that sort of thing. And I've been curious actually about the relationship between the developed world and how our immune system kind of functions in our relatively clean, hygienic world, and then how the immune system in the developing world is actually experiencing very different things that are probably a little more related to our evolutionary history. So there come the worms. That's right, helminths are worms, and this always makes people feel squidgy. I get it. But helminth infections are common in the developing world, right? That we have indoor plumbing, that we have different ways of managing waste that then do not expose us to helminths in the developed world. And pregnant women or women pre-conception get helminth infections and don't get treated for them. So in some different experiments, so in populations in Kenya and Gabon, maternal helminth infections, so they had different kinds of liver flukes or different kinds of lungworms, not great, but increased tolerance in their offspring to a later helminth infection and changed how their T cell population was sort of the stoichiometry of their T cell population, that they tended to be more T helper cells. And the main hypothesis sort of behind this is that parasite survival is completely dependent on evading the host immune system. So if your goal is to evade the immune system, you want to signal that all is well to the immune system and reduce its signaling. So anti-inflammatory signaling in a low controlled dose of helminths may actually be helpful. Counterintuitive, but we're gonna try to go with it. So we've been maintaining helminths in my lab. So we've been working with rat tapeworms. So they are the Hymenalepis diminuta, the rat tapeworm. And in this parasite, its primary host is the rat, and the secondary host, the intermediate host, is the adult grain beetle. So basically, when a rat has an adult tapeworm in their gut, the tapeworm lays eggs, and the eggs are in the feces. The feces must be consumed by a beetle, and then the beetle, the eggs develop into larvae inside the beetle, and then the beetle's dissected and the worms are given back to the rat. In the wild, this happens because rats and beetles live in and around trash and consume a lot of the same things, including each other. But what that also means in the lab is that I can infect mom and she has no way to pass this infection or inoculation on to babies. So we can dissociate how mom and baby and their experience with tapeworms might alter some neural signaling or microglial function. So just knowing that we have a way to dissociate these two particular sets of inoculations. And then I'm looking at this in a neonatal infection model. So I'm doing this with a really well-characterized model that allows me to explore what happens in a model where I give them a live infection. So this is where E. coli bacteria come in on postnatal day 4, and the whole litter is treated with either phosphate buffered saline subcutaneously or E. coli, a live E. coli infection. And then about a decade and a half of literature showed that that within 24 hours, IL-1 beta gene expression goes up in the hippocampus 24 hours post E. coli injection. And that when you look at adults, so this is in adults and we're looking here at percent freezing, and this is on a fear conditioning task. The black columns received PBS, phosphate buffered saline, as babies, and the white columns received E. coli as babies. Then when we give them a second immune challenge, so LPS is the cell membrane component of gram-negative bacteria. This is specifically from E. coli. When we give them a second immune challenge, it causes a memory impairment. That there is robust freezing and then a drop in freezing in essentially what are called 2-hit animals that received E. coli as babies and LPS as adults. Later work in my PhD showed that at the time of this LPS injection, IL-1 beta protein in the hippocampus goes way up in those animals compared to the other 3 groups. So that IL-1 beta is at least one of the cytokines, inflammatory cytokines, signaling molecules within the hippocampus that is causing this memory impairment. Follow-up work tells us that if we give mom helminths, so here in this upper graph, we're looking at relative gene expression, and on the y-axis, we're looking at mom treatment. So mom either got saline, just a control, or she got 4 helminth larvae, 4 tapeworm larvae, 3 weeks before she even conceived pups. These treatments are related to the pup treatment, so what the pups got on P4, and then we euthanize them on P5. In that way, we can see that 24 hours after E. coli in a sort of control group, they have robust IL-1 beta expression that is attenuated in the pups born to moms who had helminths. So something about mom's helminth infection seems to be reducing this neuroinflammation. And then in adulthood, we see the same pattern as previous, that the adults who got E. coli as babies and LPS as adults have impaired freezing that is rescued when both mom and babies got worms. So in this study that I'm showing here, mom and baby got worms at different times. Mom got them before baby was even conceived, and the pups got them at weaning before they were moved to their own cages. So the combination seems to be very effective at reducing a kind of lifelong inflammatory tone in the brain. Then I started here at NKU and kind of got going on the work that I've been doing with my undergraduates. So here we have the same groups, except we are not giving any weanlings, any pups, worms. We're just looking at mom worms and asking then what happens to her pups, then unfettered, essentially. Like once we've given them E. coli, how do they look across both development and in adulthood? So the first thing, the thing that has taken a very long time, but my students have been really great at it, is doing, we did immunohistochemistry for the IVA1 antibody in neonatal brains. So they sliced them at 20 microns, mounted them directly to slides, and we stained them, and then they counted exhaustively in the neonatal hippocampus in 5 slices. The exhaustive counting also required them to categorize every cell they ran into in the counting frames as round or amoeboid, stout, so a cell body with only 1 or 2 very thick processes, with thick long processes, so multiple processes with a still sort of thickness to them, or the final morphology, which is most like the adult morphology, a smaller cell body and very thin processes. The thought is that these microglia, these have moved in during the embryonic period and then are proliferating within the brain. So these are mesoderm-derived cells. And then from there are going to change morphology as they become what some people have called more quiescent, so that they express inflammatory or immune signaling molecules at much lower levels as the pups age. So hot off the presses this week, same as Melissa, exciting things happened. My students finished counting all of the postnatal day 1 brains. So we sliced brains for postnatal day 1, stained them, and they counted many brains and found this pattern of expression, which is similar to previously published work in Sprague-Dawley rats from 2012, that there are more stout cells than any other type, and there are almost no thin long cells. So no cells with thin long processes, almost none, some, but almost none. What's interesting is if we zoom in on the thick long cells, so the third type that are moving their way toward maturation, the helminth born pups, so the pups born to moms with helminths, have fewer thick long cells than the saline pups. Stick with me. When we look at P4, which is also something that we finished like right before the holidays, We have another main effect of cell type. So they have almost no amoeboid cells left. They're all either stout, so having some process bearing, or they've moved on to thick, long with slightly more elaborate processes. And we're growing in our number of thin, long cells that look more quiescent. But if we zoom in on those thin, long cells, again, we see that pups born to helminth moms have fewer of these than the pups born to saline moms. I'll tell you right now, I don't know what it means yet, but it seems like having helminths means that mom's anti-inflammatory tone or helminth related tone is slowing down the maturation of these microglia. I mean, we're only talking probably 24 to 48 hours of delay, but that can be a big deal when microglia are responsible for doing things like synaptic pruning. So I think that's going to provide a lot more questions about what we want to look at next. Maybe Dr. Barget can help me stain some spines. We can count those too. So we'll see. I'm excited about what these data might mean. The other thing we've been working on is testing in adults specifically related to behavior. So I use a paradigm called context pre-exposure facilitation effect fear conditioning. That mouthful basically means that the following happens. On day one, the rats are carried in a specific box from their home cage down to the fear conditioning room and put in the fear conditioning chambers. They get to explore for, at first, 5 minutes, and then get taken back to their home cage for 40 seconds and return to the fear conditioning room and are in the box for 40 seconds. And that's repeated one more time. So they get a total of 3 exposures for a total of 6 minutes and 20 seconds. And nothing negative happens to them in the fear conditioning room. They just look around. They're inside the chamber, they're walking about, sniffing things, nothing bad happens. They come back to the colony room, we give an injection of either saline or LPS. This is lipopolysaccharide again. And then we let them go to sleep, have a nap, do some consolidation. And the following day, we bring them back in the exact same box we carried them around in the day before. We put them in the fear conditioning box and immediately shock them. And we pull them out, put them in the box, take them home, that's it. And then on the final day, we look at freezing. So freezing is the most common response for rats who know they're trapped and who also are prey animals, right? That their fight or flight is actually a freeze and stay still. So it's the easiest way for us in this particular paradigm to separate consolidation from association from memory retrieval, right? That we can get at different kinds of the, basically different parts of the learning process and the memory retrieval process by having it separated on these multiple days. My undergrads, who have been just kicking butt, have tested 152 animals on this paradigm in the last year. So these data are a little complicated, but just to let you know that we're looking at percent freezing, and this is actually relatively low freezing for a lot of these groups. And these results are giving me pause about what this might mean right now. But if we zoom in a bit and we look at percent freezing and we collapse across neonatal treatment, and we just look at males and females overall and whether they were given saline or LPS as adults. Females injected with saline froze less than males injected with saline and females injected with LPS. So they're really not freezing very much at all. We also see that if we collapse across sex, that rats that got PBS as babies, that LPS seems to increase freezing. Whereas if they got E. coli as babies, it doesn't much matter when we collapse across sex what they got injected with as adults, they kind of freeze similarly. There's some other data we recorded with rearing and grooming at the same time. And those are showing some patterns, but the amount of rearing and grooming that they do compared to freezing is much smaller. So I won't show those data here. The other thing that's been interesting is that there is no effect of maternal helminths on any of this behavior. It does not matter. So something's going on in the neonatal period that is not making it all the way to adulthood. I'm curious, my next step is going to be to test a cohort born to naive dams. So just moms who don't get treated with anything, breed them, inject their pups with either PBS or E. coli, and then divide the groups up into pups that get worms at that time point or not. And ask the question, is the adulthood behavioral response that we saw in an earlier paper completely related to getting helminths at the time of weaning rather than mom helminths? So that'll be the key comparison at this point. My undergrads are going to keep counting microglia. So at P5 and P7, we have some PCR done and some ELISAs done at the neonatal time points. We have more to do. And then we need to do molecular testing and immunohistochemistry in adults. I'm also curious and have mentioned in my R15, which is getting submitted for the third time in February, other behavioral tasks that are hippocampal dependent but not aversive. So more exploratory tasks like context object discrimination or novel object recognition or location. So I want to acknowledge my many funding funding opportunities from Kentucky Embry. So my startup award and my idea awards. Our Center for Integrative Natural Sciences and Math provided me with some funding for some of this work, and the Faculty Benefits Committee, and then my many students who have moved rats around and counted microglia over the last 4 years. So I'm happy to take any questions.
Great, thanks. Anybody else? I have a question. I was just wondering if this, when you do the IVL beta 1 staining, I mean, is this something, the microglia, that's something very specific to hippocampus, or is this throughout the brain? I mean, I'm curious why the focus on hippocampus. Is that because it's a very specific infection or something?
Right. Well, so the hippocampus is definitely related. I mean, I think then the focus on the hippocampal dependent learning later in life and the other types of learning. So when you look at tone learning on fear conditioning, there's no impairment of any kind. So amygdala dependent learning doesn't get altered by E. coli infection.
Oh, okay. Yeah, because I just kept thinking about amygdala. I thought toxoplasmosis. It's like,
sure. Yeah. and I think so. We also looked at, I mean, I left it out for clarity's sake, but we looked at the prefrontal cortex, the cortex immediately dorsal to the hippocampus, the amygdala. Yeah, those 3 regions, and we don't see that same IL-1 beta protein spike in those locations in adulthood either.
Oh, okay. Okay, cool.
Yeah, but and at this point, there's actually more microglia in the hippocampal area and then along the ventricles than there is in the rest of the brain. Now by P30 and P60, they're just basically everywhere. But in the early stages, because they came in essentially at the choroid plexus and then moved along the corpus callosum a lot before they spread into the parenchyma.
Yeah, okay. Okay, cool.
Yeah.
All right, let's see. I'm going to try and, anybody else have any questions?
Yeah, I have a little question, Lauren.
Yeah.
How sick does it make a person or a rat to get these tapeworms in them? And is it going to be possible to, if this would work, treat humans with tapeworms?
Sure.
If they wanted to?
Right. So an uncontrolled infection, right, that you acquire naturally by eating or stepping in something definitely would make you very sick. So I give them specifically only 4 at a time. So they look the same. I mean, that their behavior is the same. We've done anxiety testing, motor, like locomotor behavior. They're the same. Those moms don't care.
And they don't lose weight or anything?
No, not at all. And they have the same size litters. Their pups are the same size because they get this infection before they even mate. Now, there are people using not rat tapeworms, but other kinds of worms, other kinds of helminths. So there was a study in MS patients where, I mean, at least 75% of the patients saw either an improvement in symptoms or a shrinkage in their lesions or both with a helminth treatment. Now they were trying to normalize the helminth treatment to like give an even dose per person rather than looking at weight or age or, you know, so, but yeah, there are a fair number of, there's also one out there about looking at, oh boy, I think it was related to vitamin deficiencies and giving people helminths and helping them with the vitamin deficiencies. That one I haven't read in a long time. So yeah, they're doing it as clinical trials, and then some people self-medicate. Not great, but in general, yeah, I think people are a little bit optimistic, but I think the ultimate goal is to figure out, are there particular molecular targets that we could kind of distill down into infusions or targets that don't involve the squeamish factor of actually ingesting worms.
Okay, now I'm sorry, I'm glad you guys are all sticking with us. A little behind. And Simran, you had me so confused. I was pulling up your PowerPoint and then it came up. I was like, what am I doing? Okay, well, so you're good.
You know, this is what, when you are working with on a Mac, it like sometimes like, so I had to figure it out because I wasn't able to share even, you know, earlier one day, but I thought it was just, I didn't like pay much attention to it. So I was like, okay, this is,
we're all good. Okay.
And I just wanted to check everybody's okay with me. Like, I think we are already like, you know, okay, good. So I'll go ahead. And so like, you can see my title here is Legionella pneumophila. And so my title slide went off, but I'm going to start with Legionella pneumophila. So this is a gram-negative bacteria. It's found in warm waters, and basically it is able to cause Legionnaire's disease. And this is a severe kind of a pneumonia, which mainly occurs in immunocompromised individuals. In healthy individuals, the bacteria just causes what we call as Pontiac fever, which is a flu-like infection. And it's really, sometimes people don't even know that they had this. And so basically how this bacteria, it's a bacteria that lives in water in association with amoeba. And so it forms these biofilms, and it basically is going to be present in the biofilms. And from there, and this is like air conditioning units, cooling towers, spa, all these where there is this circulation of air, the contaminated water, when it is in a free form, when it's inhaled, it passes to the lungs, and that's where it's going to set up an infection. And so now I have another problem. Yeah, finally more. So how does it grow? So just to kind of give you an idea, it's an intracellular bacteria. So it grows within the cell rather than other bacteria, you know, which like to grow outside of a cell. And so, and interestingly, it also likes to grow into the macrophages. And so alveolar macrophages, when they inhale the bacteria, now the default process is anything when it's inhaled by, you know, or engulfed by the macrophages, to be more precise here, it basically will get endocytized. It will, you know, fuse with the lysosome, and as a result, it would be destroyed here. However, with Legionella pneumophila and various other intracellular bacteria, they are able to prevent this phagolysosomal fusion. And so you can see how they do is that within 5 minutes, and here are some electron microscope pictures, within 5 minutes or so after they get engulfed, they will attract these secretory vesicles, and then these vesicles will surround them. And in 30 minutes, you can see all these you know, secretory vesicles from the ER and from Golgi, they will surround it. And in another about 2 hours or so, this whole vesicle, it'll form like a ER-like structure. And so this is called as a Legionella containing vacuole. And then within this vacuole, this will grow. So the main here is like, again, the electron micrograph, and you can see these are the vesicles that are attached to this. And then in about 10 hours or after about 8 hours, it begins to replicate. So 2 hallmarks of the infection are that it inhibits the phagolysosomal fusion, and then it's responsible for the ER recruitment. So how does this happen is, so the main virulence of the bacteria is dependent on this type 4 secretion system, which is called as a DOT ICM. So DOT is for defective organelle trafficking. ICM, they basically mean the same thing, and same, it just 2 different labs almost at the same time had identified them. And so we have detailed those, and so we call them DOT/ICM secretion system. And so what is it is made up of? There are 2 regions in Legionella that basically form this transport structure. And this transport structure then releases effectors or proteins that are produced in Legionella during an infection. And so here you can see this transport system is really important. And so we use as our negative control, or we say DOT A mutant, which is this region here, green, and that gene. When it's mutated, the transport system is not formed. And in this graph, you can see this is DOT G that has been used here to, the same thing. You can see here that the mutant, when it lacks DOT G, it will not be able to grow within the macrophages. So what does this DOT ICM transporter do? So this is what the transporter will eventually form, a structure like this. And so these are all the proteins that have been formed within the bacteria, which only will be released once the bacteria makes a contact with a macrophage or a protozoan cell. In the environment, it does the same thing with the protozoa. And so when there is a membrane to membrane contact, then it releases the DOT ICM will then release all these effectors into, and so all these effectors go into the cytoplasm, and they then regulate or like manipulate various processes within the host. And some of the very well-defined is the vesicle trafficking from ER to Golgi, the phagolysosomal fusion, ubiquitination, which is very, very currently one of the main topics that, several researchers, including my PI, has been doing, apoptosis, and then protein inhibition. So CDF was the very first effector that I began to start working in, back in 2003. And at that point, we had no idea what the effector was this effector was doing. And so what we identified eventually was this effector. It blocks apoptosis or kind of delays apoptosis of a cell. So macrophages, when they get infected, right, either destroy the pathogen, or if they're not able to destroy pathogen, they will undergo apoptosis so that they, are destroyed. And as a result, the pathogen is also eliminated. So what this bacteria does, it doesn't want that to happen. So there's kind of a tussle here between the pro-death signals that the host macrophage will produce and the Legionella, because it's an intracellular bacteria, needs to survive within the environment. And so it's going to produce some pro-survival signals. And so this is where CDF came into picture. There was another one around the same time, the CID-HA. So cell death, we believe, is mainly as a result of the macrophages. So to understand more as to what is going on in here, and if there is something else, how the bacteria really is able to multiply and understand various effectors, what we did here was that in our lab, first this is during my postdoctoral time, we wanted to see how many effectors are produced by this bacteria, because different labs were giving information that, okay, they have discovered this and this effector, and we were learning about a lot of effectors. So this bacteria, it has about 3,000 genes. And so what we did was we basically did a kind of a throughput screening where we cloned all the hypothetical proteins that was, over 300 base pair into, first we wanted to check whether they can be translocated during an infection, that is, that they can be secreted. So we did a beta-lactamase assay, and we found that over 300 effectors are actually translocated into the host cell, which is really a lot. It's like 10% of this bacterial proteins that actually go into the host. while we were doing this, we also started to look at what was the role of these effectors. So we used yeast cells and tried to see if they could produce any kind of a growth defect phenotype in the yeast cells. So Saccharomyces cerevisiae has been even a model here. And then we wanted to look if it inhibits the growth, if it's lethal to, the yeast. So this was a project which, of course, I published on this one. And then there was I took upon another one where I wanted to just look where does these effectors localize within the cells. So just used a very simple approach where I tagged all the proteins with GFP and then looked for their expression. And then eventually, if I saw something interesting, something different, then I stain them with the appropriate antibodies. Like, for example, if they appear to be on the mitochondria, then I stain them with the mitotracker. If they appear to be on the cell membrane, then I used appropriately. And if they were somewhere else. So to my surprise, which was not by that time known much, was that they were proteins that actually localized to the nucleus. So there were 6 proteins that actually went to the nucleus. And sorry about this. Okay, so there are 6 of these proteins that actually localize into the nucleus. Now, at that point, of course, we now know this is now becoming pretty common that there are effectors that are produced by not just Legionella and other bacteria also that go to the nucleus and are involved in epigenetic modifications. But at that time, we were kind of surprised why these proteins were going into the nucleus. So we thought maybe it was just an overexpression thing. So we, and maybe GFP somehow was taking them. So we used different ways. Of course, in this particular slide, you can see this is a GFP, and here these are the flag tags which we use to confirm, and then this is stained here. You can see all the cells that are expressing the flag tag proteins. Those are the ones that are here. This is the DAPI staining, and you can see the co-localization directly onto that. So so this is the hypothesis that I've been working then, is that Legionella pneumophila effector proteins that translocate to the nucleus are involved in transcription regulation of the host during an infection. Now, when we say during an infection, we have to first confirm that they are secreted into the host during an infection, and they go to the nucleus. So this was the very first experiment where under infection conditions, so basically here, I used a wild type Legionella pneumophila, and then I also used a .A Legionella pneumophila, which does not have a transporter and therefore will not be able to secrete. That's just like a control, negative control. And so you can see here all 6 of them. And so basically I did an infection of the macrophages, mouse macrophages. And then after 24 hours of infection, I got the nuclear fractions and collected the nuclear fractions and the cytoplasmic fraction and probed them for the presence of these proteins. They were all flag tagged just so that it would be easy for us to do the detection, because we couldn't really have antibodies for each one of them. And so you can see here, in the nuclear fraction, all of these proteins during an infection do go into the nucleus. And so this is a cytoplasmic fraction, and these are just the appropriate controls over here. So once this was established, we wanted to know, okay, do they have any role to play in the intracellular growth of the bacteria? Now, one thing with Legionella is that because it produces 300 different effector proteins, if you delete just one effector protein, it doesn't do anything. It just nicely survives, and it's happy there. So as expected, I remove any one of that protein. I have no defect. I removed 2 of them. I decided to do a combinatorial deletion mutants. So I deleted 2 of them, still did not see any defect. And then when I deleted 3 of them at a time, at any given time, and it could be any different 3. And so then I begin to see the defect. And so you can see here, these 2 are the defects. So typically, in 72 hours, you see that the bacteria grows like 1,000-fold. And these could only grow about 50 to 100-fold. And so there is a defect in them. And when you add, complement in the gene, even any one of them, it basically grows like a normal LPO2. That is the wild type strain. And so this is basically, okay, so only if you, 2 or 3 or more probably now. So now we are in the process of constructing quadruple mutants. We basically have all of them, just have to verify them. And then we would see how much growth defect they have. Well, meanwhile, I wanted to know, okay, if these are going to the nucleus, then nuclear trafficking is not something normal. Within a cell, the nucleocytoplasmic trafficking is highly regulated. And so what happens in a normal host cell is that there is these are all GTPase processes, like the GTP binding proteins. And so there are importans which will bind to the cargo. So this is the cytoplasmic side here. And here is important and cargo. They bind to each other. And these importans basically bind to the cargo through specific sequences called as NLS sequences. And so we would started look if these NLS sequences were there, which will enable these proteins to then bind to the importans. So when I looked for the nuclear localization signals, the only way, because all the software that are available, they really don't predict any NLS sequences for these proteins. So what I decided to do was like literally break the protein into half. So if this protein LNE A is 546 amino acids, this was like 275 amino acids, put onto one end, which was like the N-terminal end, and then the other half, which comprised of the C-terminal end. And then basically looked for whether it still goes to the nucleus or doesn't go to the nucleus. And so you can see here, LNE A has the N-terminus going to the nucleus, whereas all of these here will have the B, and then back again, the leg AS4 has a N-terminus. And then on the basis of that, I basically reprobed and found that these are the sequences that probably match or appear to be the NLS sequences. So, okay, so if these are NLS sequences, then they have to also bind to the importance. And so here is where I look for the interaction of the Legionella nuclear proteins with important alpha. So there are different importance here. There's alpha 1, alpha 2, alpha 3, which are involved in this trafficking or, taking the cargoes from the cytoplasm to the nucleus. So basically here I have the GFP constructs. I have importance that I put, tag them with the flag tags so that I could do immunoprecipitation assays here. And then using HEC293 T cells, after 24 hours of transfection, carried out an immunoprecipitation assay. And you can see here that not all of them are binding to importance. Only, so this RUV-O is RUV-Q is another protein which is toxic, and we are working on that one in a different project. But LNE-B here and LNE-A, these are the only ones that basically bind to the importance. The others did not really seem to bind to the proteins, even when they were expressed really well in a cell. And so this made us think, okay, so maybe some of them are using the host, transport system, but not all of them are using the host transport system here. So, you guys were talking, we did a lot of work after this to find out, okay, if these proteins are toxic or they do something good. So all these apoptosis assays, cell death assays, MTT assays, looked at their phenotypes carefully, did some, everything came out negative. So we finally decided to do, since, okay, we believe that they are involved in epigenetic modifications, some differential gene expression analysis using RNA. And so here we just simple RNA extraction after, so this is still under overexpression conditions using the GFP construct. We did the RNA extraction and then, RNA sequencing. So now we have some RNA sequencing data. So this was all done with the genomic core at UL. And so here you have this pipeline that, that was used for analysis. So what we see here is, you can see this is a summary of how many genes were expressed or differentially expressed. And so they were in seg B, they were total of 57, or which is basically seg 10, it's just my labeling, 57 genes that were differentially expressed, of which 47 were upregulated and then 10 were downregulated. RUFS O has the same pattern, 41 overall, and 35 are upregulated, 6 are downregulated. Now, in these two proteins, you can see the MERK-E and LNE, this is LNEP, we didn't have a lot of changes. Now, these are not because there were not really any changes. These proteins have a low expression, and that's why we believed in the beginning they might be slightly toxic, although we never found anything with them, like they were, killing the cells or there was any cell death processes going on. So these have low expressions upon transfection. So they're only about 30 to 40% of transfection. And so here basically what you see is like we right now here don't have a flow cytometer, and that's why I actually wrote an MRI grant for that, because I was like, we need to have a flow cytometer to separate these cells. And the same problem also happens with the infection, because not all the cells get infected all the time. It's only 10 to 30% of the cells that get infected when I'm doing infection studies. And something like a flow cytometer will completely change the game for us, because then we can enrich those infected cells and then study them more carefully. So here is some initial sequencing data that I have, and it kind of implies what our hypothesis is. So this is with respect to seg10, and you can see that a lot of, although the p-values are not that great here, a lot of histone modification, chromatin modification, acetylation, all those are the processes that are happening. And so now what we are trying to do is we are actually looking at that, and what we have planned is some RT-PCR studies and see what is going on. So we're basically focusing on that with respect to these pathways. Similarly, in case of a RUB-O, what we saw that it has something to do with the interleukin biosynthesis. And so I'm not trying to kind of cut it short and not going too much in detail, but if you see here, you come back to the cytokine biosynthetic processes, cytokine metabolic processes, a lot of that. And so it looks like it's something to do with that process. So same with this project, we are trying to now focus on those lines and really see. So we're going back to the data and looking exactly what is happening there, how many fold changes is there, and then looking in that pathway, how we can, make sense of this data and then use it. So here is the overall summary. So there are 6 different effectors that we are basically working on that go to the nucleus during an infection. 2 of these we know that they use the host nuclear trafficking pathways for the, for transport. We don't know what do the other 4 do. And so that would be interesting to know, how do they get into the nucleus? Is there any Legionella protein that is acting as an important or behaving as an important? And then we see that the triple mutant, like if we delete about 3 or more, maybe possibly mutants, these effectors, then we are able to see the growth defect, the intracellular growth defect. None of these are toxic to the mammalian cells. And now we know that the RNA sequencing data kind of implies that it is involved in epigenetic modifications. And I would like to, thank you, my students. So you can, all of them have contributed and done all the experiments that failed and, gave us no results, but they were really done. And so my PI, Dr. Zhaoqing Luo, who is now totally into ubiquitination, bioinformatics score has helped us a lot. And I myself have learned a lot of RNA analysis now, which I had no idea about. And of course, funding from, K-Brain. I land up saying K-Brain, KY in Brain still takes a while. And so from that was, I had, startup grant from them, and then I've had 2 idea grants. So I'm really grateful for that. Last year, Genomic Core helped with a lot of RUB-Q, which is a toxic protein. So we did some RNA analysis of that, and we have some very interesting data on that too. And of course, I had initially NSF EPSCoR to get a microscope in my lab, and of course, a lot of funding, internal funding. So thank you very much, and happy to answer any questions if you still are, with me.
Thank you. Yeah, that was really interesting.
And I'm so sorry about all the mess that was happening in the beginning.
Oh, no, that's
You
and I'm
didn't see
sorry I
my
didn't keep
mess
us on track.
at the beginning, Simran. I
Oh
was
yeah,
trying to
so that's what
do a
my
thing.
computer has been giving me a problem, and I'm trying not to do any updates. But problem with Mac is you update and then something will change. And so, but I had to restart it, and that's how I missed the starting for you.
You can always watch on YouTube.
Oh yeah, that's what I will do. Yeah. Any question for me?
Any question?
I guess I've got
Lauren
a
has-
little. Oh, I've got a little bit of a question by Lauren about what you said at the end about none of the proteins that that go into the nucleus are toxic. So I'm trying to get my head straight. You showed us with some of the triple mutants that some of them don't grow as well in the macrophage. Oh, there it was down. If you go down.
Yeah.
That it takes knocking out several of these to get an effect on growth compared to wild type that are living inside macrophages. Yes. Okay. So they're not toxic as evidenced by what means, I guess, would be one question.
So here, what we are talking in this particular case is that we are actually growing the Legionella, and then the Legionella strain, the wild type strain, lacks those effector proteins, ability to produce those effector proteins, right? So those are our deletion mutants. Now, when we are doing these kind of studies here, these are under overexpression conditions where we are using mammalian cell lines, like basically HEC 293 cell lines, not necessarily macrophages. And again, the reason for not using the macrophages is because the transfection rates in macrophages are not very great. And then again, we would have a same problem if we are not able to separate those cells, by some means, like, by fax machine or something, we really will not be able to see much impact. So that's why we use the 293 T cells, which are very easy to transfect. Now, when we transfect these cells, what I meant was not toxic is that to these cells, they do not show any kind of toxicity. Like they do not prevent their growth or their,
Roundup or refractal, whatever. Yeah.
Yes.
Yeah. Okay, so it doesn't necessarily, you've got a lot of lovely data here, a lot of good work, and I'm just trying to see if it's, consistent with, well, if they're going to the nucleus, they're doing something to the nuclear gene expression, but they're not hurting the cells. That's what I was thinking of when you were saying they're not toxic to the cells. And I see now that's a different kind of assay. And I mean, hopefully you're working with these nuclear effectors that are doing something that's either damaging, as you say, to the host cell or helping the Legionella survive.
Yeah, so what we are believing is that definitely they are doing something that is helping Legionella. Now, in that process, they don't have to be necessarily, damaging the host cell.
Right.
So they can just prevent some, events or some signaling to happen in the host and then still help the Legionella, but not be. That's what we are currently, at least our data is showing us. Okay. Now, we do have, like I said, there is a protein in this category, where we saw that it was toxic to yeast cells. And then when I transfect that particular protein into 293 T cells, the HEC 293 T cells just stop growing. They don't grow. And they don't die either. So basically it's just, and same thing happens with yeast. The yeast doesn't grow at all. And so that particular protein is like, maybe I'm comparing it in terms of that protein and saying the word toxicity, but that one is really toxic and it won't allow the cells to grow, and it will just, cells over time will die.
Cool. Like I said, a lot of neat stuff you're finding out lately.
Yeah.
Thanks.
Thank you.
Very nice. Well, I'm sorry, I didn't keep us on track at all, but it was just because everybody's infectiously excited.
Yeah, Simran had the yeast, she had the growth, she had no worms though. Oh, well.
Yeah, I didn't have any worms. Yes.
Don't
That was
tell Lauren.
interesting though. Yes.
All
Well, thank
right,
you so
thanks,
much
everyone.
for waiting, and thanks.
Okay, bye.
Thanks,
Thanks,
Martha,
everyone.
for arranging
Bye.
this. Nice to see everybody.
Good to see everybody. Yes.